After explaining the anatomy of dicot root, monocot root, and the differences between the anatomy of dicot root, monocot root, and anatomy of dicot stem, monocot stem, and the differences between the anatomy of dicot stem and monocot stem, and the anatomy of dicot leaf, monocot leaf, and their differences, and secondary growth in dicot stem and secondary growth in the dicot root. Let me explain the remaining part, that is in the 6th chapter of the first PUC, anatomy of flowering plants. So, branch of biology deals with the study of tissues called histology. So, plant tissues broadly classified into meristematic tissue, permanent tissues, and epidermal tissues. Meristematic tissue composed of cells which are in the state of active division. These cells undergo both periclinal and anticlinal divisions. Cells which are in the state of active cells are closely packed without leaving intercellular spaces. Contains nucleus, dense cytoplasm, and cell wall. Thin cell wall composed of cellulose, that is very important point. There are certain cytological features which enable meristematic tissues to undergo rapid divisions. That is the presence of thin cell wall, and they respire rapidly, releasing energy, and they contain monomer of mitochondria and K+ ions are the important characteristic features of the meristematic tissues. Meristematic tissues further divided into the apical meristem, intercalary meristem, lateral meristems. Apical meristems present at the root and stem tips, and intercalary meristems present at the base of the internodes. These are the nodal regions, and this is present at the base of the internodes. And lateral meristems are present towards the sides. During the formation of the leaves and the other branches of flowers, sometimes the meristematic cells stay back. That develops into axillary bud. Later the axillary bud develops into a branch. So based on their location in the plant body, position in the plant body, apical meristems, intercalary meristem, apical meristems present the stem and root tips and cause elongation. Intercalary meristems present at the base of the internodes and cause elongation, and the parts which are affected by the grazing of the animals can also be once again that part can be produced due to the action of intercalary meristems, and the elongation of the leaf and the elongation of the pinus needles, it is chiefly because of intercalary meristems. Lateral meristems are present towards the sides and they increase the girth of the plant body. Increase in girth due to activities of lateral meristem known as secondary growth. Example, cork cabium and interfascicular cabium. I explained this. And these cells undergo dedifferentiation, having lost capacity of divisions. Once again, they regain the capacity of divisions, and they help in wound healing process, that is the lateral meristems. And coming to the permanent tissues, they have lost the capacity of divisions. They are of two types: simple permanent tissues and complex permanent tissues. Simple permanent tissue composed of only one type of cells. Example, parenchyma, parenchyma, sclerenchyma. Parenchyma cells are isodiametric cells. That is very important for your NEET examination. They have thin cell wall composed of cellulose. That is very important, and they leave lot of intercellular spaces between them. So that is called parenchyma. Storage, secretion, and photosynthesis. That is the parenchymatic cells which contain chloroplast called mesophyll help in photosynthesis are the important characteristic features. And they also in erenchyma they help plants to float on water. They help to maintain the buoyancy in case of aquatic plants. That is about the parenchyma. Coming to the parenchyma, thin walled cells but corners thick. Corners thick because of the presence of cellulose, hemicellulose, and pectin. This is very important. Cellulose, hemicellulose, and pectin also present in the corners in case of parenchyma, and they offer mechanical support, and the cells are closely packed without leaving intercellular spaces. Especially these are present in the petioles of the leaves and growing part of the stem, especially parenchyma present in the hypodermis of the dicot stem and bundle sheath extensions of the dicot leaf, and especially in the petiole. So next, the sclerenchyma hexagonal cells are thick-walled and composed of lignin, a chemical called lignin. Sclerenchymatous cells are of two types, sclerids and fibers. Fibers are elongated structures. Sclerenchymatous fibers are associated with xylem called xylem fibers. They are also called wood fibers. They offer mechanical support. Sclerenchymatous cells, fibers associated with phloem called phloem fibers are bast fibers. Bast fibers. They offer mechanical support. That is, the phloem fibers, they are present in jute, hemp, etc. So this is about the sclerenchyma. Sclerenchyma consists of sclerids and fibers. Sclerids have lignin and a cavity called lumen. These sclerids are present in the grit portion of or the pulp portion of the fruits such as guava, apple, pears, sapota, and the seed coats of leguminous plants or pea plants. Once again I will repeat, sclerids, very important. They are present in the pulp portion or grit portion of the fruits such as guava, apple, pears, sapota, and the seed coat portion of the leguminous plants, that is the pea plants. That is about the sclerids. And coming to the vascular bundles, complex permanent tissues. Complex permanent tissues are xylem and phloem. Xylem is water conducting tissue present in vascular plants, and the xylem consists of primary xylem and secondary xylem. Primary xylem consists of protoxylem and metaxylem. If protoxylem facing towards the pith portion, it is called endarxylem present in stems. If the protoxylem facing towards the periphery, called exarxylem, that is present in roots. And this xylem is a complex permanent tissue. It consists of various components such as the tracheids and vessels. Tracheids show the tapering ends and elongated structures, and vessels short and stout, no tapering ends, and both they are composed of the lignin deposition, and they do not contain nucleus and the protoplasm. So xylem components are tracheids, vessels, xylem parenchyma. Parenchymatous cells associated with xylem called xylem parenchyma. Storage of food is their main function. The extensions of xylem parenchyma, though not given in the NCERT book, 2 times asked in the NEET, the extensions of xylem parenchyma called tylosis. They enter into the sapwood, converting the sapwood into hardwood. They block the sapwood, converting the sapwood into hardwood. That is the role of the tylosis. The extensions of xylem parenchyma called tylosis. And xylem fibers are the hard fibers. They are present in the leaves of agave, banana, canna, and offer mechanical support. Coming to the phloem, phloem is food conducting tissue. It's a vascular tissue present in vascular plants. Phloem, primary phloem and secondary phloem. However, the primary phloem get crushed in case of the monocote stem, and primary and secondary phloem get crushed because of the production of the extensive secondary xylem during the secondary growth in a dicot stem. And phloem is complex permanent tissue composed of more than one type of cells, such as sieve tube elements, companion cell, phloem parenchyma, phloem fibers. Sieve tube elements are rectangular in shape and they have sieve plates. Sieve plates are perforated with the sieve pores. So these sieve pores, cytoplasm of one sieve tube element is in contact with the cytoplasm of adjoining sieve tube element. Cytoplasm chiefly consists of sucrose and P proteins, amino acids, that is P proteins, nothing but phloem proteins. Sucrose is the composition of chemical composition of the phloem sap, that is the cytoplasm of the phloem. That is very, very important. And this is the boron which help in the transportation of carbohydrate. Carbohydrate transportation takes place in the form of sucrose, that is the disaccharide, that is called cane sugar, and that is the composition of this. And during winter season, the sieve pores get plugged because of callus formation. Associated with such sieve tube, and these sieve tube elements, one more important characteristic feature for the NEET is they are devoid of nucleus. No nucleus but living. That is very important. Even though there is no nucleus, but it is living. That is about the sieve tubes. Associated with the sieve tube, there are companion cells. Companion cells contain nucleus and it controls the activities of sieve tube element. And one more important point is companion cell establishes connection with the sieve tube elements through pit fields. In case of gymnosperms, no sieve tube elements. In the place of sieve tube elements, sieve cells are present. Associated with the sieve cells, there are albuminous cells. That is very important. Albuminous cells and sieve cells. Phloem parenchyma. Parenchymatous cells associated with phloem called phloem parenchyma. Storage is their main function. However, phloem parenchyma absent in the monocot stem. And sclerenchymatous fibers associated with phloem fibers, they are also called bast fibers. Xylem fibers called wood fibers present in agave banana canna. Phloem fibers are present in fox, hemp, jute plant. So that is about the phloem. And coming to the vascular bundles, the arrangement of vascular bundles. If the vascular bundles are arranged in the same radius, they are called conjoint. Same radius, not radial. Here there will be a confusion. If they are arranged in the same radius, they are not said to be radial, but they are conjoint. If the vascular bundle xylem and phloem alternates with each other, arranged on separate radii called radial. Radial can be seen in roots and conjoint can be seen in both monocot stem as well as the dicot stem. That is also important point to be noted. And then next to this, epidermal tissues. Epidermal is composed of tangential elongated cells covered with cuticle. That is very important point. The continuity of the epidermis interrupted by stomata. Stomatal apparatus consists of two kidney-shaped guard cells. Each guard cell contains a nucleus and a chloroplast. Inner walls are thick and outer walls are thin and elastic. Inner walls are thick, and these guard cells are surrounded by the subsidiary cells, and there is opening is called stomata. That is very important. And then next to this, trichomes are present in the dicot stems. Such trichomes are absent in the monocot stem. So this is about the other than anatomy, that is the anatomy of dicot root, monocot root, dicot stem, monocot leaf, secondary growth, and the remaining aspects that we have completed. Thank you.